Hello. I'm going to talk you guys through the protein synthesis lab a little bit. Okay. So just because it's really easy to get confused because it's not difficult, but it's easy to lose your way. Okay. All right. So all of the directions are basically on this first page and then kind of overflowing into the second page. Okay. So if you get to the point where you're like, wait, what am I supposed to do next? In order. Okay. So I'm just going to walk you through. Okay. And I'm not going to do the whole thing, but I'm going to kind of fake it. Okay. So, okay. Here's what we're going to do. Reading the directions. DNA is a template for DNA. Basically what we're doing on, I keep scrolling, on data sheet 1 is we are practicing our complementary base pairs. And my video is not working very well, but that's okay. All right. So let's do that. Let's practice that. Okay. So I'm going to take my handy dandy pen here. Right. And so by now, I hope you know about the complementary base pairs. So remember, the bases are the parts of the nucleotides that line up in the center of the double helix of DNA. Right. So you're welcome, Pickle. So my daughter's in the pool with a snack, so she's happy. Okay. So you're gonna, basically the rules are this: A's and T's go together and C's and G's go together. Okay. So super easy, right? If you have an A, what is complementary to that? What matches up with that? That's right. And if it's a T, it's an A. I think I said it backwards already. Whatever. Anyway, if it's an A, that's right, a T. If it's a C, you see where this is going, right? First step will take you a minute and a half. Okay, so that's step 1. Check. Okay. Transcription. Now, transcription, you've watched videos about this, you've read about this by now. Okay. Essentially what you're doing is you're taking 1/2 of the DNA helix, one of the strands, and you're building a complementary strand of RNA that matches the DNA. Essentially, the complementary, the base pairing rules are the same, except RNA doesn't have T's. It doesn't have thymine. Instead, it has uracil. Okay. So now I'm on data sheet 2. Okay. I'm going to change my pen color so that, I don't know why, because it's RNA now instead, I guess. Okay. So okay, so now it's the same rules, but there are no T's. So where there would be a T, it's going to be a U instead. Okay, so T goes with A, so that's fine. A, normally you'd write a T there, right? But it's RNA, so you can't. So it's going to be a U. And then C is going to be G. G is going to be C. A is going to be U. T is going to be A, so on and so forth. Okay. All right. Now, yeah, transcription, constructor complement, blah, 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 blah. Check. Okay. Now, it says bracket the codons along the length of your mRNA. Each codon is 3 letters long. Use the genetic code chart found in your textbook or handout. It will allow you to associate codons with the correct amino acids. Okay, so let's do that. So codons are essentially your 3-letter words of DNA or RNA that code for a particular amino acid. Amino acids, remember, are the building blocks of proteins. So, right? So we got to separate out our codons. So you can do little brackets like this, or another option is you can just sort of, what I do is I just kind of underline 1, 2, 3, every third one kind of big like that, so that I can, just so I can see them really clearly. Okay, so let's see, where were we? Back to the, see how I'm flipping back to the instructions? Bracket the codons along the length of your RNA. Each codon is 3 letters long. Use the genetic code chart found in your textbook or handout to allow you to associate the codons with the correct amino acids. Okay. I'm going to put a check even though I'm not done with that yet. So where am I going to find one of these genetic code tables? There's one in your book. There's also one, what page is this? Page 7. On page 7 of this handout, right? And so what you're doing here is you're looking at this table to figure out what the codon codes for. So they're called codons because it's a word that codes for an amino acid. All right. Pickle, I'm making a video. Stop pestering me. I'll be with you shortly. Oh God, I went too far. Okay, so my computer's all irritated. Okay, so I'm just, because I'm flipping back and forth, I'm just going to memorize this. Okay, so AUG is the first one, and then CUA. So put that in your head. AUG, CUA. You can print this out so that you, if you want, right? Absolutely, you can always print it out. So it's easier. Okay, so AUG was, where is AUG? AUG is right here. And what was the other one? Was it CUA? I think it was CUA. Okay. So I'm finding those on my little table. Okay. And what's next to each one of these codons is an amino acid. You do not need to know the names of the amino acid. You can just use the abbreviations. Okay. So MET stands for methionine, but it doesn't matter. Just call it MET. Okay. Also, you'll notice that next to MET, it says start. That's because for any strand of mRNA, the start codon has to be first. It is where translation starts. Okay. This is really easy for me to remember, by the way, because my birthday is in August. So AUG, that's when I started. So therefore, it's the start codon. Okay. So that's my first one. And then my next one, right, CUA codes for leucine. But like I said, you don't need to know the names. You don't need to memorize this table. You just need to know how to use it. Okay. And it's in a circular form in your textbook. So it's kind of in a different format, but it still works the same way. Okay. So use whatever works better for you. Okay. If you want to look up, genetic code on, Google it, right? You could do that too, and that'll be fine. So what were they again? It was MET and leucine. Okay. Okay. Now notice what I've done here is I put some little arrows and they don't line up perfectly. Dang it. I need to fix that. I did not fix that. I'll fix it hopefully before I post this. Okay. So this little arrow is supposed to be going from the mRNA to the amino acid. And what that's telling you is it's saying, okay, for AUG, the amino acid that codon codes for, you're going to write on that line. So that's MET. And then CUA codes for leucine, so on and so forth. Okay. You will notice that some, are you talking to Luigi? Is he prowling? Yeah. Okay. You will notice that these, whoop, not that one. Okay. Try that again. You will notice that these stay stop next to them, right? So MET was start, AUG is start. Those 3 codons are stop codons, which means that there is no matching amino acid that goes with those. So you stop. There's no tRNA, there's no amino acid that goes with them. Okay. I just want to point that out so you can see that. Okay. All right. Okay. Now, what do we do next? Go back to the instructions. Okay. Where were we? We were, right? So it says use the genetic code chart found in your textbook or handout. Allow you to associate the codons with the correct amino acids. Done. Okay. Then you're going to make some tRNAs. Oh my gosh, you guys, my daughter is so cute. She's asking me like deep science questions like, why is the wind air? After this, I'm gonna get in the pool with her and explain it. Anyway, she has the best timing. Okay, so the next step says cut out 21 tRNAs. I gotta take these off. Okay, so this is a tRNA. Okay, and so there's a sheet of them on page 7 of this packet. Is it 7 or is it 8? It's there. Okay, burp, burp. It's page 8. Okay, so these are them. So you got to cut them out like this. It says you need 27 of them. Now, in a perfect world, not 27, 21, I can count. In a perfect world, you all have printers and you can print these out and then cut them out and do this, okay? But I do appreciate the fact that some of you don't have printers. So what you could do, if that's the case, is you could put a piece of paper over your screen and trace them, right, on regular paper, and then cut them out and do the whole thing. If you absolutely don't want to cut them out, you don't have to. You can kind of fake this part, but I really recommend you don't, because I think that it helps you kind of understand this process, right? So the whole point of this lab is for you to do some kind of hands-on transcription and translation, okay? All right, so it says cut out 21 of your tRNAs, your transfer RNAs, and then it says write one anticodon on each. Let's talk about what an anticodon is. So an anticodon, as its name implies, is the opposite of a codon. Okay, so what do I mean opposite? Well, what that means is that it's going to be complementary to a codon. Okay, now we'll talk more about that in a second. Okay, so here is a list of the different anticodons that you're going to make. And this is where you need your paper clips. Now, I'm assuming that you all got the same color spread that I got in your little bag of paper clips. You are not going to need all of these, by the way. So you're welcome for some free paper clips, I guess. Okay. So the colors that I got in my package, they were in a little box, okay, were yellow, pink, red, white, green, and blue. Okay. This requires us to have 2 additional colors. I wrote in purple and black, but you can, if you want to, change that to something else. So like if you're like, well, I have silver paper clips at home, you can change purple to silver, right? Or another option is you can color some of the white paper clips. So I took a Sharpie. I don't know if you guys will be able to see this. I took a Sharpie and I cut there. You can tell that they're white, but I put black on them, and then I made a purple one too, right? Because I had a Sharpie. So you can do that, however. So you can do them as purple and black, or if you want to use different colors, like if you're like, you know, I happen to have some rose gold ones at home, right? Just, you know, cross it out and change it to whatever color you're using. Okay. Okay, it doesn't really matter as long as you put it on here so I can see how you changed it, right? So it's clear to me what's going on. Okay, now the other thing is it's really important that you write on this correctly, okay? So this is important. This is the main way that people screw up. And you might want to do this in pencil just so that you don't screw up, okay? Okay, so what we're ultimately going to be doing with this is we're going to be lining it up against, I'm taking this and I'm putting it up against my screen, okay? So I'm gonna like draw one, I'm gonna draw one in so you can see how I'm holding it, okay? So it's got this part, this is where the anticodon is gonna be written, and then this is the rest of it, and this is the part that the paperclip attaches to, okay? So what I'm gonna be doing is I'm gonna be holding it up against my screen, so I'm gonna try to draw this, I'm gonna draw it in, it's gonna be bad, that's okay. I'm going to draw it in blue. Okay, so the way that it's going to be oriented is it's going to be oriented Okay, that was terrible, but whatever. Okay, but that's what it's going to look like. And so you'll notice that there's 3 boxes. So 1, 2, 3 boxes. They're not even put anyway. Okay, so the way you're going to write your codon is you want to hold this so that the pointy part is pointing to your right. Okay, and then you're going to write the letters from top to bottom. You're not going to hold it this way. You're going to hold it. Yeah. Okay. All right, so let's go back to this for a second. So my first tRNA, I'm going to draw, right? So I'm not going to do the first one because that one's 3 A's, so you can't screw that up. So let's do this one. Okay, so yes, Pickle. What happened? Okay, leave it in your, she's in a floaty and she's got a bottle of water and snacks. She's spoiled. Leave it right in your floaty with you, okay? I'll come get it in a minute. Okay, so it kind of looks like that. So the letters in order from top to bottom. So A, G, like that. Okay? Okay. I gotta get her out of the floaty. Don't have kids. I mean, dude, there's the best thing that'll ever happen to you, but they're also a giant pain in the butt. Just saying. Okay, where were we? All right, so you're gonna make 21 of those, okay? And like I said, you want the letters going from top to bottom so that the pointy part is pointing towards your right side when you're writing on it, okay? Then each one, you're gonna add the correct paper clip, which represents a building block in the protein called amino acid, okay? So my AAG one, it says yellow, so I'm gonna get a yellow clip and I'm gonna put it on like such. See that? Okay, so it should, I didn't write it on here. I will, let me do it. What was it? AAG. So for that first one, it'll look like this. AAG. Okay, so you're gonna do all of those, right? So you're 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Okay. So you're going to make all of those, all 21 of them, and then you're going to just like stash them off to the side. Okay. Now we're ready to, we've done transcription. Now we're ready to translate. Okay. This should say codons, not condons. Okay. So what you're going to do is you are going to go back to data sheet 2. We're on data sheet 2. Right? And we're going to match the anticodons with their codons. Okay, what does that mean? So anticodons, like I said, are essentially the opposite of a codon. Okay, so let me just erase that thing so that it's not in our way. So what would be the anticodon that matches that codon? Well, complementary base pairing, right? So AUG, what's the opposite of that? Mommy, you're so strong, Ellie. Okay, so it's it's illegible, but it's UAC. Okay, so you will have made one of these that has UAC on it. I did not, because I just made the one. But when you make yours that says UAC, right, from top down, your UAC is going to be purple or whatever. I'm going to turn this one into a UAC just real quick. Okay, UAC. Dun, dun, dun. Okay. So then what I'm going to do is I'm going to hold this up against the screen or your piece of paper or whatever. I'm going to line it up. Okay. Then I'm going to get, and I'm going to like hold it there on my piece of paper. Okay. Then what I'm going to do is I'm going to get the next tRNA that has the next. Not right now, Pickle. It's not on right now, baby. So then you're going to use you're going to figure out what the next anticodon is that matches this codon. So remind me again, what does anticodon mean? Anticodon is the opposite. So you're going to find your little tRNA that has GAU. Right? And as you go, you want to write in your paperclip colors. So my first one was purple. Okay. And then your next one, GAU, I can go and look it up. But really, you're going to actually have it on paper. Is pink. GAU is going to be pink. It's fine. Okay. And then you're going to have these two tRNAs next to each other. And let's go back to the instructions. The amino acids create a peptide bond via dehydration synthesis, and they link together. You know how to link paper clips together, right? The paperclip chain. You take the one paperclip, you take the other paperclip, you link. Then this tRNA, the one at the top, goes away, and it can be reloaded and used again later, just like in real life. Okay. Your other tRNA is still attached to this one, so let's fake that. Okay, and that's growing. Yeah, and then you're going to figure out what your next anticodon is, bring that tRNA, link the amino acids, take the next. So you're going to gradually build your paperclip chain, because each paperclip is an amino acid. You're going to build a protein, and you're going to be keeping track of the colors of the paperclips as you go. Okay, right here. Okay. You're going to keep going until either you reach the end or you reach a stop codon. Okay? Hold on one sec. So many interruptions. So many. Okay, where was I? So you're going to keep going until you hit a stop codon or you reach the end. Or maybe it'll be both. Okay. All right. Then what do we do? How do we know what to do? Well, we go back to the instructions. So it says, continue this process until all the codons have been matched with the tRNA and the amino acids have all been linked together. A three-dimensional functional protein occurs when the white amino acid is separated from the yellow amino acid by 3 amino acids. So I'm just going to fake this because I don't, whatever. So I made a chain. Dun, dun, dun. This is not right. I'm just faking it for your benefit. Okay, so let's say this was the chain I made, right? It says a 3-dimensional protein is made when my yellow and my white are separated by 3. So here's my white, here's my yellow, 1, 2, 3, perfect. So I'm gonna link those together. Like that. Dun, dun. Okay, and then you can lay it down on your table and you can draw it where it asks you to do that. You can draw it, or you can take a little picture of it with your cell phone and slap a picture on there. Whatever you want to do is fine. Okay. You can come back to my school one day, but not until the virus is gone, okay? Okay. Okay, so where were we? All right, sketch the protein paperclip at the bottom of data sheet 2, or attach a picture, whatever you want to do is fine. Okay. Now you're going to do, essentially, you're going to take it apart and reload your tRNAs with the correct paperclips, right? Going back to this, okay? And you're going to do this whole process again 2 more times with a couple of mutations, okay? So data sheet 2 is your like normal protein, okay? Data sheet 3, as you'll see from the directions, is we're substituting one base for another. So notice in this little blank here, what was this before? It was ACC right there, and now it's A blank C. So it tells you to switch it to a G, I believe, in the instructions. Let's go back and check. Yes. So all you're gonna do is you're gonna change that to a G instead of a C. You're gonna do all the same stuff. Now you'll notice that most of this isn't going to be any different. In fact, really, the only thing that's going to be different is this, because nothing else has changed. Everything else is going to be the same. So the only codon, and therefore anticodon, that's going to be different is that one. Okay, so fill in the whole thing, right? But the only difference you should see comparing this to data sheet 2 is right in there. Okay, and then you're going to follow the directions like you did before. So if, you know, once you make your chain, whatever it looks like, it's not going to be super different, but it probably will be a little bit different. Okay, but just like before, if your yellow and your white are separated by 3, then you're going to link those together, and that's what you're going to draw. Okay, so you're going to draw that. Boom. Okay. Now, the second mutation that we're doing is on data sheet 4. This mutation is more significant because what we're doing now is we're doing a insertion. There is another typo. I'm finding typos like crazy, okay? So what we're doing is you are, the way that I like to describe this, so we'll talk about, this will also be in the video that I haven't recorded yet about, you know, mutations. So insertions and deletions, what's happening basically is like you have this strand of nucleotides and you basically are shoving something in, and what it does is it pushes everything down. We call those frame shift mutations because what you're doing is you're shifting the reading frame. Okay, so I think the easiest way to understand this is if you've ever bought something at drugstores, really like to do this, where like a display is spring loaded. So there's like lipstick or deodorant or whatever, and it's in this like little like spring loaded thing. And if you take one out, the next one pops forward because it's spring loaded, right? Well, essentially that's what we're doing here. We're either taking something out, or in this case, we're inserting something in, and it's going to push everything after that down. Okay. So instead of just substituting something, right? So after where it says CTG, it then before said, so originally it said CTG, and then it said AAA. Well, we've been instructed that we need to add a G before the 19th base. So where it originally said AAA, you're now shoving in a G right here. And what that's going to do, so I'm shoving in a G right there. And what that's going to do is it's going to push everything else forward. And what it's going to end up doing, because we've inserted only one base, is it's going to change your whole reading frame. So now, right, it was CTG and then AAA, but now it's CTG and then GAA and then ATA. Yeah? Okay. So the top will be the same because you didn't mess with anything at the top from the first time. It's only going to be at this point after that it's different. Okay. And then do the same thing, right? So the process of filling out this is the same. The process of making your paperclip chain is the same. If you've got a white and a yellow separated by 3, you link them. If you do not have a white and yellow separated by 3, you do not link them. Okay. you answer questions about this stuff. All right. So here's the little genetic code thing. It's gonna be, there's 9 per page, so 2 pages and then a couple extras. Okay. Right, if you want to trace them, you can do that. The last 2 pages of the lab, this is totally just bonus for fun if you want. Not 2 pages, the last 3 pages of the lab. Like I said, not required, just fun if you want to. Okay. It's kind of fun to make DNA. And so these are all the parts of the DNA, and you can actually build a DNA molecule, right? So if you're feeling like I need to do something hands-on so that I can appreciate the different parts of the nucleotide and how they fit together, and then, you know, that's what this is for. And it's kind of fun to color each thing a different color. So for example, notifications, right? So I'm coloring all my phosphates pink. Oh my gosh, this is faster than real coloring. It's amazing. You see the moon, Pickle? And then I'm gonna color all the, so anyway, just pretend I did all of them. And then I'm gonna color all, oh wait, no, yellow. Cool, Pix. I'm gonna color all my thymines yellow. And if I'm cutting them out, I don't need to color in the lines because I'm cutting them out anyway. And then just for shiggles, I'm gonna color my adenines. Anyway, you get the idea, right? So you color each thing a different color, and then you cut them out, and then you build it. And if you need guidance, ask me for help. But like I said, this part is totally optional. It's if you want, just for fun. Okay. What you're ultimately going to turn into me is pages 2 through 6, I think, because 6 is the questions, right? So page 6 is the end, and you're going to turn in. If you make any changes to your colors, turn in page 1, 2, just so I know what you did. Or just starting at page 2 to 6 is what you're turning in. So you can just write directly on the Word document. You can print it and take pictures and whatever. Okay. I think that's clear. If you have questions, ask. Okay. Okay. I'm done now.